Hi, everybody. Good morning. We're going to get started. Dr. Lombroso has an extraordinarily rich presentation. Dr. Lombroso has a very rich presentation, so if everybody could get seated, we're going to start right away. A very short introduction by me. Dr. Lombroso is a professor at the Yale Child Study Center in New Haven, Connecticut in the United States. He has a very interesting career because he's not only a child psychiatrist, but he's also a molecular biologist, and he's going to speak to us about how we learn and how we don't. My only comment as a trauma psychiatrist is that in the field of trauma, there are no neurobiology centers of just memory. They're memory and learning. And in our field, we often focus on the traumatic memory, but not the learning part. And that's really much of what this talk will be directed at. Thank you. Welcome.
Thank you very much. It's a pleasure to be here. I want to thank the organizers, Professor Caffo, for inviting me. Apologies to you if you went to the wrong room. But in any case, today what I will be doing is talking to you about learning and how we learn and how we don't. And I have a couple of points that I want you to leave here with. One of the concepts that I'll be talking about extensively throughout today's talk is this of the concept of synaptic plasticity. Okay, so when we have to learn, the spines at the end of our neurons have to change shape. And that's one of the concepts that I'll be telling you about, and that concept is synaptic plasticity. And you may have also heard that in order for synaptic plasticity to occur, we have to have what's called long-term potentiation. And I want to explain to you exactly what this term also means. And it's required for any formation of long-term memories. You must have long-term potentiation. Finally, throughout my talk today, I'll be reminding you of the relevance of the work that I'll be discussing today and various psychiatric disorders. I do this in part because I am a molecular biologist, and I believe very strongly that it's important to try to understand some of the molecular events that precede psychiatric disorders. And so that's what I will share with you today, some of the parts of this story. So learning is typically divided into declarative memory and non-declarative memory. Declarative memory is what you can declare, what you can talk about. And non-declarative memory are memories that you are usually unconsciously learning. Okay? And I wanted to test you a little bit on these very basic concepts. So this individual is... Now, I know from a reliable source that Ernesto Caffo used to have a poster of her. Yes, it is indeed Sophia Loren. He had a poster of her on his wall, right? Yes. In addition, we have all seen Michelangelo's David. The question for you, though, to test your declarative memory is, when was it made? Okay, within 50 years, when was it made? There we go. I heard 1502, it was actually 1504, but that was close enough. Okay, so those are examples of declarative memory. And the capital of the state that I come from, Connecticut, is Hartford. That's not a test for declarative memory. What I wanted to tell you is a story, though, of an individual in a nursing home who still lives in Hartford. His name is H.M. And H.M. is probably one of the most studied individuals in all of neuroscience. And that is because when he was about 7 or 8 years old, he lived in a town just north of where I live called Middletown. And he was riding his bicycle, and unfortunately he was not wearing a helmet. He fell down and had a traumatic injury and developed a seizure disorder. Initially, the seizure disorder was successfully treated with medications, but over time it became progressively more and more severe. and medications no longer worked. He was unable to attend school, and he was unable to keep his job. At that point, he went to visit a Dr. Schofield. He was a neurosurgeon, also trained at Yale. He did his work at Hartford Hospital, and he wanted to try a new experimental surgery, which was, in those days, this is the 1950s, they were removing epileptic foci. Okay? And that's what he wanted to try to do, and that's what H.M. had. He had, when he had fallen down, this is, of course, the brain, and this is the temporal lobe, and if we turn over the brain, this structure's here, at the bottom of the temporal lobe, shown here, and are the hippocampus. Okay? And this is because they sit right on the skull. When you fall down and have a traumatic injury, it's this part of the brain often that is injured. And so Dr. Schofield decided to remove the sites of the epilepsy. Okay? And that's the result. They removed through suction the hippocampus and some of the nearby structures to remove this region here, the hippocampuses. And the surgery was successful in the sense that his seizures were much improved. However, what happened at this point was that H.M. lost all of his declarative memory. He was unable to form any new memories. So over the course of the next few weeks and months, he was unable to remember anyone who came into the room, unable to recognize Brenda Milner, who was the psychologist who was invited in to study him. And Brenda Milner decided over the next several months, she did a number of tests that I now want to go into, which helped to clarify the difference between declarative memories that H.M. was no longer able to do and non-declarative memories that he was able to do. So she did this simple experiment, and let me spend a moment just explaining it. If you go home tonight, you should try it. Here is a star with a border. When you go home, just try to, with a pen, stay in the border. Okay, relatively easy psychological test. Let's make it a little bit more difficult now when you go home. Try to do it by only looking through a mirror. Okay, so you can't look at your hand anymore, but you have to look through the mirror at what your hand is doing. A little bit more difficult. And there was an apparatus in which they did it. And how do you expect you would do over the next 4 days if you were here in Florence practicing this test? You would make initially a lot of errors, but slowly but surely you would improve in your ability to make fewer and fewer errors. How did H.M. do on this test? Did he get better or did he not? The answer is yes. I wouldn't be telling you otherwise. He had exactly the same type of improvement in his non-declarative memory. However, remember that he had absolutely no memory of the test. Every day Brenda Milner came in, he didn't know who she was. He had no memory for the test itself. She had to explain the rules again of what he was trying to do. So this is an example of non-declarative memory. You have no memory or no conscious awareness of how it is being formed, but it is being formed irrespective. So just to summarize at this point, H.M. was unable to form declarative memories, but had no deficits in his non-declarative memories. And there are many examples of non-declarative memory. I mentioned here tracing a star. It's a motor skill that requires a particular part of the brain called the striatum. Playing the piano, I know Helmut Remschmidt is not here, but some of you may have had the pleasure of listening to him at times playing the organ, and you can see his hands flying over the keyboards. But when he starts to learn a piece, his declarative memory is engaged, and over time, and many of you play an instrument, you realize that your fingers all of a sudden start learning the piece without having to look at the music, etc. Riding a bicycle is another example. Many of you can get on a bike these days and just take off. But how you actually, your muscles learn to actually ride that bicycle is a form of non-declarative memory in which the striatum is involved. I just mentioned pasta as a silly joke because in America, of course, they give us spoons to eat pasta because they know that we can't learn how to. But for the rest of the talk, I'm going to go to several other topics. The first will be habits. Habits, of course, I know many of you have automatic, or rather cars which have shifts, and you know that as you drive that car, you are unaware of, just your ears hear the sound of the engine and you shift that car into the next gear. And there are many examples of habits that you're not aware of learning, but you do learn them, non-declarative memory. And fears, we'll be talking quite a bit today about fears and how they get formed. The relevance of this part of the talk will be as we discuss phobias, panic attacks, and anxiety, and post-traumatic stress disorder. Let me just summarize then this first part of my talk. Long-term memory is typically divided into declarative memory and non-declarative memory. Declarative is the memory of events and facts. The restaurant that you went to last night, the smells in that restaurant, and the taste of the food. All of these are examples of declarative memory. And the hippocampus is critical to the formation of these types of memories. I do want to just make one point here, is that the process by which you turn short-term memories into long-term memories is called consolidation. Okay? You consolidate short-term memories into long-term memories. Okay? And that is what requires synaptic plasticity. Now, the hippocampus is required for the consolidation of memories, of declarative memories, but the memories themselves are not stored in the hippocampus. So, for example, if you did go to a delicious restaurant, the sounds of that over the last few days, the sounds would be stored in the auditory cortex. The tastes that you had and remember in the gustatory cortex. And the visual aspects of that experience are stored in the occipital cortex. But if you remove the hippocampus, that event, that consolidation cannot take place. Non-declarative memory, and I'm just showing three examples here, procedural or motor skills, the ability to learn how to play the piano, ride a bike, or do the star example, that requires various parts of the striatum. Classical conditioning, and we'll be spending some time talking about this, the amygdala, cerebellum. Drug abuse is an interesting, we won't have time to talk about drug abuse, but the current ideas are that this is a form of non-declarative memory, that the synaptic plasticity that occurs in the striatum and the nucleus accumbens that you're not aware of is one of the reasons that we develop drug abuse. This is just a note to myself to take a drink of water. We're moving to the next part of the talk. I don't know if you noticed, but I was bending down when that happened. I still can't look at it.
Tell me how old you are.
I'm 17.
You're 17, okay. And tell me about your symptoms.
I would just, sometimes out of the blue, I would just kind of start feeling like really scared, and my heart would like start beating really fast, and I'd get out of breath and stuff, and just sometimes I'd get like a choking feeling too.
Okay. How long would that usually last for?
It depended sometimes. Sometimes it'd last 5 minutes, sometimes 30.
Okay.
It's just different every time.
Okay. Was there anything that seemed to provoke that?
Some things would, like if I just started thinking of something that kind of made me nervous, that would kind of do it, or just it'd happen out of the blue a lot. too.
Okay. When did they first start?
We first started noticing it when I was like in 7th grade, but now that we think back about it, I had them when I was like little too, but we just didn't know what they were.
Okay, and do you remember much what they were like when you were younger?
I would kind of have like this feeling in my throat sometimes, you know, that like I had like a big lump in my throat or something, or I just like get really nervous and just start, you know, like breathing like really fast and stuff for no reason.
Okay.
Panic attacks. Post-traumatic stress disorder and phobias, anxiety disorders. My proposition today is that these disorders are caused by deficits and problems in how we form memories. Okay? It's a hypothesis. A great deal of research is going on in trying to test this hypothesis. And what I'd like to do now is to move on to my second phase of my talk, which is to tell you how you actually study these types of questions in the laboratory. So you have a clinical question in mind, and you want to test it experimentally. How do you go about doing that? The amygdala is in part the region that we will focus on today. And I have to explain to you what we actually do in the laboratory to test fears. And fear, it's called fear conditioning. And we typically take an animal, in this case a mouse or a rat. I missed that. Let me just go back for a second. So we take a mouse and put them in a particular room or chamber. Or we give a sound, and immediately that at the end of the sound, they receive a very mild shock. And they learn very quickly that the sound is associated with something noxious. Okay? You only have to test them or train them 1 or 2 episodes. And now the question comes of when do we test them? We can test them first at 1 hour, or we can test them later at 24 hours. At 1 hour, we call that short-term memory, and at 24 hours, it's the consolidation that requires synaptic plasticity that has gone on. And what happens at both 1 hour and 24 hours when you, in fact, do put them, for example, in the same room, these mice or rats will freeze. Okay? Or if you give them the same tone that they heard earlier, again, they will freeze. Okay? And that's an indication that they have learned that the tone or the room means that a noxious stimulation is about to happen. Of course, we don't give them the shock here. We just measure their ability to freeze as an indicator of their having learned, okay, and consolidated the fear conditioning. Let me just show you an example. Here's, in this case, a rat. Listen for the tone. This is being tested at 24 hours later. Has it learned? You see the freezing? Okay. Key concepts here that I want to talk about. Long-term memory requires, as I've been saying, synaptic plasticity. New synapses, those connections between neurons, have to be formed, or older ones have to be strengthened. And these actual structural modifications that occur to synapses require protein synthesis. So when I talk about spines, these are the structures that you can see here. These are the points of synaptic connections. So neurons outside will make synaptic contacts on these spines, very typically. This is a medium spiny neuron from the striatum. And in red here are actually these contacts that are being made. A typical neuron will have 10,000 spines and will have 10,000 synaptic connections. And so what we're going to talk about now is how the modifications occur to these spines as learning gets consolidated. Okay, we have to have structural changes occurring. And another key idea here is we don't want all of them to change. We want a subset, a very small group of these spines to have a morphological change. And how does that happen? Okay, so what I'm going to go through very quickly now, we're moving into a little bit of molecular biology here. Growth factors, and here I'm showing one of them, BDNF, or synaptic activity itself, are required for this synaptic plasticity. And here is the problem. We want a signal, let's say a growth factor. A signal comes to the outside of the neuron, and somehow the signal has to get inside into the nucleus, and we have to have new protein synthesis. So I mentioned to you that synaptic plasticity, the morphological changes that occur to spines, requires new structural changes, and that requires new proteins. So very quickly, let me go through this. I'm going to use growth factors as an example. This process is termed signal transduction. The first event is that the growth factor will bind to a specific receptor. In this case, it's a BDNF receptor. The BDNF receptor has two ends. One, extracellular, binds to its ligand, and at that event, the binding of the receptor to its ligand causes those receptors to come together or dimerize. When that happens, a very rapid event occurs. A phosphate group is added. That's called phosphorylation. It's added, in this case, by the enzymatic activity of the receptor itself. It cross-pollinates cross-phosphorylates itself. So we have these phosphate groups now, and the addition of a phosphate group to a protein is very often a way of activating that protein. Okay? So what happens, these phosphates act as a magnet, and they pull out of the cytoplasm various proteins, and a cascade is initiated. Various kinases, those are proteins that add phosphates, okay, those activating groups to proteins, and a group of transcription factors get phosphorylated. Transcription factors are master control proteins, okay? So they rapidly move into the nucleus where they sit on the genome and initiate transcription. mRNA or a message is made. That message rapidly comes out to the cytoplasm, is translated into a protein, and that protein is now used for the synaptic plasticity that I talked about. So just to summarize, a signal arrives, goes into the nucleus, transduction, messages get made that are needed at that very moment for the synaptic plasticity. In this case, it's an ion channel that's being added to the membrane. One of the key kinases, I mentioned kinases, the group of enzymes that add phosphate groups, is called ERK. It's an extracellular signal, so it responds to extracellular signals, okay? And it's regulated by those extracellular signals. It turns out that this protein, ERK, is a key regulator of synaptic plasticity, and we'll come back to that as we go along. I now want to try to define long-term potentiation. Very simply, long-term potentiation means that when a neuron receives a synaptic input, the synaptic output from the postsynaptic neuron is strengthened, okay, and strengthened very strongly for a long period of time. Let me show you with an example. Okay, here we have an individual learning or smelling a rose. If we could, in the cortex, record from a neuron that is receiving input from this smelling of a rose, we would have a very typical response to that smelling. Learning has not occurred yet. Okay, now let's learn something. Let's associate by repeated pairing of the smell and the sight of that rose together. Okay? So we have those two events occurring at the same time, and now let's record from this neuron. Something has happened right here. After multiple responses, this postsynaptic neuron now responds much more strongly to either the sight or the smell alone. This change here is called potentiation, okay? And it lasts for a long time. That's why it's called long-term potentiation. And you can do it the other way around. Initially, if you just have the smell of the rose, a very low level of response, but if you repeatedly pair the smell and the sight, something happens at this postsynaptic neuron to strengthen the response to either the smell or the sight of that rose. And this is called long-term potentiation. How does this happen? Oh, just a final point that is very specific. If you instead smell an onion, it does not do the same thing because it has not been paired if you paired it initially with the sight, yes, you perhaps would get a faulty memory. Okay, what happens then in fear conditioning? Where does long-term potentiation occur in fear conditioning? And for this we have to travel to the amygdala. So this is a part of the amygdala here. This region here is called the lateral amygdala, the medial and the ventral. Here's the basal lateral. And this structure here is the central nucleus, a key player in the consolidation of memories and the output that I'll tell you in a second. So initially, the mouse experienced a tone and a shock. They came, in terms of the tone, into the ear, and part of that signal went to the thalamus and then was transmitted directly to these neurons in the lateral amygdala. At the same instant, and this is the critical point, you have to time it. At the same time that the animal hears the tone, they have to feel the shock. And so the shock also goes to the thalamus and comes to those neurons in the lateral amygdala. And this is where long-term potentiation then happens, so that if 24 hours later you only give the tone, these neurons now respond very strongly. Okay? Consolidation has happened in these neurons here. And a signal then is passed to the central nucleus, and from the central nucleus it goes down to these various deeper structures. Okay? So and the response there is, for example, an increase in heart rate, urination, the blood pressure that increases, etc. And you can see the response here. The response says there are typical signs and symptoms of fear and anxiety. What I want to do now is to go even deeper into the events at a molecular level that happen for the changes that we've been discussing, the long-term potentiation and the synaptic plasticity that occurs. This is, let us pretend, a neuron in the thalamus that is sending its axon here into the lateral amygdala. These are mitochondria. These circles here are the vesicles. Let's pretend that this is a glutamatergic neuron. The release of glutamate into the synaptic cleft rapidly moves across the cleft and binds then to specific receptors, and the specific receptors here are in the postsynaptic terminal. Now what we're going to do is we're actually going to go on a little plane trip into the cleft itself. Okay? I think we can all fit. Okay, we're getting there closer. Okay, so here, what I wanted to give you the sense of here, this is the presynaptic terminal, and here is the postsynaptic terminal. These are receptors that are sitting, waiting for glutamate. Now, there are three ways that what we've been talking about is long-term potentiation. Okay? How to increase the response of the postsynaptic neuron to the presynaptic signal. There are three ways that researchers have looked at this. First, you could simply increase the amount of glutamate that's released. Okay? Increase the release of the neurotransmitter. More neurotransmitter per action potential that arrives. A second way that you could do this is increase the number of receptors. The number of receptors that are present. So you could add more receptors to the postsynaptic membrane. Or, finally, you could change the response of those receptors so now they stay open much longer. Okay? They're much more sensitive to a particular neurotransmitter. We call that, we're increasing the open probability. They stay open for a longer period of time. It turns out, this is just to say what I just said, so you can increase the amount of glutamate that's released, increase the response of each of those receptors on the surface, or actually put more receptors on the surface. And I'm saying it yet a third time in another figure here. We can increase the amount of glutamate that's released. Here I'm showing two different types of glutamate receptors. One is called the AMPA receptor, otherwise known as GluR1, GluR2, and the other is the NMDA receptor. These are the receptors that respond to glutamate by binding to glutamate. So one way we could increase postsynaptically the response is by increasing glutamate. A second is by phosphorylation. I mentioned phosphorylation earlier. When you add a bulky phosphate group, it changes the conformation or the shape of the protein, and this receptor now stays open for just a slight second longer. Okay, so the open probability has increased. Or you could actually increase, again by phosphorylation, receptors, and now that event, the phosphorylation of these receptors, leads to a greater number of receptors on the surface of the membrane. Let's look at this a little bit more in detail. Here we're seeing an action potential coming down and the response of the postsynaptic neuron, and now we're going a little bit closer to the membrane so you can actually see what happens. There's a group of receptors over here. They are constitutively recycled, okay? What we're interested in is these receptors, and what will happen is when that first signal arrives, a great deal of an increase in the surface number of these receptors occurs, and now when we get the signal again, the response will be much stronger. Boom. Okay? Something has happened postsynaptically, and the response to the same signal is much stronger. Okay? Long-term potentiation. Okay? And these events that I showed you just in this cartoon are part of the process that we call synaptic plasticity. Okay? And these events happen when two signals arrive at the same time to a particular neuron. David, Michelangelo. Finally, somewhere in my brain, a neuron finally learns it. Okay, again, let's go back to key concepts here. Long-term memory requires synaptic plasticity. New synapses are made, old ones are strengthened. It requires the structural modifications that I've been talking about, and structural modification requires protein synthesis. You've heard the term long-term potentiation, the way by which a postsynaptic neuron responds much more strongly to a presynaptic signal. And I reintroduced this protein called ERK, because that was, you remember, the kinase that is required, okay, required for these events to occur. It's required, here I'm showing one of its jobs, which is to rapidly phosphorylate transcription factors, which initiate the process that we know of as a translation of proteins and protein synthesis. In all parts of the brain, going back to this summary slide from earlier, in all parts of the brain where memory happens, whether it be declarative memory or non-declarative memory, this protein, pERK, must be activated. Okay? And if it's not activated, you don't consolidate memories. You don't learn. So, if that happens, it's not surprising that a number of developmental disorders occur with a disruption in this particular molecular pathway. Okay? We have talked a lot about ERK. Here's ERK, right here. And ERK itself has to be phosphorylated, but this pathway from growth factors or from synaptic activity converge on this protein here, ERK, which then moves rapidly to phosphorylate proteins within the nucleus, and they in turn initiate what I'm showing here. This CREB is a transcription factor, and you have the initiation of gene expression proteins or genes are transcribed and proteins are now translated. So let's look at a couple of disorders in which this pathway is disrupted. Neurofibromatosis, tumors, okay, and cafe au lait spots are classic presentations of this disorder. Many of these individuals also have tumors inside of the central nervous system, and in the past, in fact, it was thought that this was the reason that they had 50% of them had cognitive deficits. That's not true. The reason these tumors may cause them to have a seizure disorder, but not the cognitive deficits. The cognitive deficits come because this pathway is disrupted. Let me show you how. One of the key events right after growth factors bind or synaptic input occurs is this protein that is called RAS. Binds up another small molecule called GTP. Their names are not that important, except it must be very carefully regulated. And there is a protein, it's called a GTPase activating protein, or a GAP, that blocks or opposes the binding of GTP. That is what the gene NF1, or neurofibromatosis gene, is. It's a GAP protein, and it opposes or puts a brake on this particular pathway. So let's put a mutation in these genes, in this gene, and what would you expect would happen to the activity of RAS? Right, it would go up. There would be a dramatic increase in RAS activity. Okay? That's not very good thing to have. And when you have a dramatic increase in RAS activity, you disrupt this entire process that I've been talking about. And you do not learn properly, or not as well. Okay? And you have mental retardation. A second disorder, I won't spend a lot of time just to point out that Coffin-Lowry syndrome is a mutation in this protein here, RSK2. You put a mutation in this protein, and again, you disrupt this key signaling pathway by which learning occurs. Okay? If in Rubinstein-Taybi syndrome, we go a step further, and we put a mutation in a Krebs binding protein. The job of this particular protein is to open up the DNA so that the transcription factor can actually bind to it. And if we put in a mutation in CBP, we get this particular disorder called Rubinstein-Taybi syndrome. Let's go back, though, to what our central character of this story has been ERK. And now we ask, what actually regulates ERK's activity? And my laboratory has for years been studying a protein called STEP. It stands for striatal enriched protein. Striatal enriched protein. And it actually regulates the activity of STEP. In a lot of these pathways, there's a balance between activating a protein and inactivating a protein to maintain the proper level of a particular protein. I showed you just a few developmental disorders in which ERK's activity is disrupted. Okay? Here, STEP is another player in this pathway. It regulates the activity of ERK. How does it do that? Again, we go back to phosphorylation. ERK needs to have a phosphate group on it to be active. Okay? And so if you remove this phosphate group, it becomes inactive. That's shown here. So this is the protein STEP. What it's going to do is dephosphorylate this tyrosine here. And when that happens, ERK is no longer active and can no longer signal. So now, what I'd like to do in the last part of my talk is to go back to the laboratory and try to learn or show you examples of how we start to study learning in the laboratory using a particular animal model. And the animal model I've already introduced is fear conditioning. And I've already told you that this protein, ERK, that STEP regulates, ERK is required for the consolidation of fear memories. The question we asked, the hypothesis was, perhaps we could block ERK signaling. And if we could block ERK signaling specifically in the lateral amygdala, perhaps they would not learn fear conditioning. And that was the hypothesis, and now I'll show you how we actually did it. So the idea here is to make a mutant STEP protein This mutant STEP protein will bind to ERK because of a mutation that we've introduced into STEP, and it won't let ERK loose, so ERK won't be able to signal. So let me just say this again. We introduce a mutation into the STEP protein. It's an inactive STEP protein now. It still binds to ERK, but notice what happens. When it binds, it could no longer dephosphorylate, and it does not release ERK anymore. Okay, so the idea is that we are trapping ERK. We're going to put this particular protein that we've made into the lateral amygdala, allow it to enter into neurons, and see then if when we do our fear conditioning, do these animals learn? Can they consolidate this type of learning? This is how we do the experiment. We actually take rats and we do surgery because we have to put bilateral cannulas into the lateral amygdala. We then are going to let them recover, and now we're going to infuse bilaterally into the lateral amygdala this mutant STEP protein, okay? And the idea is that it will diffuse into those neurons, and we then train these animals. You've seen the training, how we do it. We give a tone, then a shock, okay, and a very mild shock. And then we test them at 2 points. The first time we test them is at 1 hour, short-term memory. Short-term memory does not require ERK activity, okay? It's only when we have synaptic plasticity that occurs over the next 24 hours that we will develop the memories, the consolidation of fear memories, and we test again then at 24. So let me just repeat. The animals should develop short-term memories for the fear conditioning. They should freeze, but they should not consolidate that memory, and they should not, when we test them at 24 hours, they should not have learned the fear conditioning. Short-term, this is a slide showing the short-term experiments. So the animals had the infusion of the TAT STEP or a control, just another protein that was it had no effect. And you can see here that when we tested at 1 hour, everyone learned. They all froze equivalently. But let's look what happened at 24 hours. So now we're testing, I'm showing you the video at 24 hours, and listen for the tone and whether the animal freezes. You've seen this before. This is the control animal. Now we're going to the experimental. Do they also freeze? They haven't learned anything. Okay? And we can quantify that. Of course, we've done many, many animals. And you can see that at each of the tones that we do at 24 hours, the controls freeze just like they are supposed to, but the ones infused with this mutated STEP protein that binds to ERK and prevents ERK signaling, they don't learn. Okay. I think that was going to be where I was going to end. We have time for questions. There's a question over here. And then there's a question over here, number 2. Yes?
Stephen Stanley from Dawson in England. Thank you very much indeed for a very impressive presentation. One of the facets of ADHD is that these young people often don't seem to respond with fear to situations other children would. Is that been looked at as whether that's part of the ERK and STEP sort of program?
No, the answer is no. People have not looked at a molecular level at what happens to ERK in attention deficit disorder. If you're asking, are there synaptic connections from the frontal cortex, which is the area suspected to be involved in ADHD, there are connections directly to the amygdala to modulate those particular neurons that we were discussing today.
Thank you very much.
There's another question over here. Right here.
Thank you very much. Can we have hope about treatment, perhaps for
This is a very good
post-traumatic syndrome?
Right. I'm sorry that I didn't go into this a little bit more. The question was, okay, so you've heard a lot about the translational neuroscience that we do, how we test models in the laboratory. But the real question is, how do we translate that into therapy? Okay? You are not going to be involved at the traumatic event of a child who is raped or sees a trauma, or a soldier who has post-traumatic, at the initiating event. Okay? You won't be there. But you will be there later on during the therapy. So the question is, it's a concept that I didn't go into, but there is now thought to be another period of time when memories return, okay, into your mind, a process that's called reconsolidation. Okay? So every time you have and recall a memory, whether it be of Michelangelo or if it be of a traumatic event, that memory gets restored, okay, reconsolidated. It turns out that the exact same paths that lead to the initial consolidation of those memories are also involved in the reconsolidation of those memories. And so that opens a window of opportunity. Perhaps you could intervene later, okay, during the period of time of reconsolidation, perhaps with medications, and people are now trying to do that, okay, to disrupt the reconsolidation process by disrupting those particular memories from being laid down a second time. So that's one area of research that's just beginning these days. And it applies not just to post-traumatic stress disorder, because, I mean, a critical part of what I was trying to convey is that the pathways in the hippocampus, the pathways in the striatum, in the amygdala, okay, in all parts of the brain, for drug abuse in the ventral striatum and nucleus accumbens, those pathways are preserved. They are the same. Okay, the consolidation of memories, whether it be of declarative memories or synaptic plasticity of drug abuse, use those same molecules in all parts of the brain. And so people are now actually in drug abuse trying to look if they can disrupt some of these events in animal models, of course. There's a question over here, two questions over here. Oh, let me just, while we're setting up, make one other point, and to connect what we've been talking about today with another disorder, Alzheimer's disorder, in which very similar pathways are appearing today to be disrupted. Okay, it's another fascinating area of neuroscience, is the disruption of cognition and cognitive events in Alzheimer's disease. And some of the same molecules that we just talked about today are appearing to be involved in some aspects of Alzheimer's disease. Yes?
I'm wondering if you can comment on the developmental regulation of this process, because one of the things I'm experiencing both as a psychiatrist and a parent is the learning potential of my young children.
Sure. So it's not an accident that learning occurs during childhood, you know, so wonderfully, and that's why we go to school. One way, or the other is to keep them out of our hair. But the process of learning appears to be accelerated during childhood and adolescence. It's not that we don't learn later on. Of course we do. You know. But it turns out that some of the receptors that I was talking about, the NMDA receptors, they come in different flavors. Some of them appear to be much more abundant early in childhood and are thought perhaps to be responsible for the facilitation of learning early on. As you grow older, as animals grow out in the lab grow older, and we as humans grow older, there's a shift away from that particular NMDA subtype into another particular subtype that is a little more sluggish and doesn't learn quite as well. And there are other reasons why that happens, but yes, there's a question in the back.
Yes. In PTSD...
Post-traumatic stress disorder, yes?
Rapid eye movement therapy is used. What do you think of that in...
Well, I mean, I should say that I know nothing about that. But that doesn't stop me. There is perhaps other people in the audience who in a second can address that, but I think that what I do know is that there is, the treatment for post-traumatic stress disorder very commonly has been extinction, exposure therapy. And it is thought that you are learning, you're not erasing an old memory, but you are learning a new way of dealing with that memory, okay? So we know that's a fact because very often, as you know so well in your clinical practice, that when you stress someone who has had a long course of extinction or exposure therapy, they can revert back to their old symptoms. So they haven't erased those memories, but it's now thought a new form, a new consolidation of memory has occurred. But whether rapid eye movement and that type of therapy is involved, I really don't know enough about it. Sorry. Yes?
We today learn about growth hormone involvement in the initiation of learning. And if I recall right, it is mainly released in the brain at night time.
At what?
Night time.
At night time.
And need sleep. So how about those people who are working shift workers and for days and weeks sometimes? Are they the same in their memory consolidation?
Well, you remember that your mother always told you to have a good night's sleep, right?
Yeah.
Right. Part of the reason we're now learning is because that's the period of time where consolidation of memories occurs.
Yeah.
So she was right. But in terms of growth hormone itself probably is not involved in this type of learning. Okay. Growth hormone is more involved in the growth of different body structures. But there are, and so that particular growth hormone you're saying is secreted at night. I don't think that the two are connected, but I do think that during the process of sleep, consolidation in fact is occurring. You saw short-term memory has to be consolidated, and that in fact does take time. Thank you.
Thank you.